Hello everyone, my name is Dinesh Kumar. I am a PhD student in the lab of Professor Charles Schroeder at University of Illinois. Today I'll be speaking about transient stretching and relaxation dynamics of anisotropic vesicles in flow. Now, there are several motivations behind studying lipid vesicles in flow. One such motivation is that lipid vesicles are increasingly being used in drug delivery purposes. Another motivation is that lipid vesicles can be used as a simplified model system to understand the properties of biological and synthetic membranes. For instance, the shape of a healthy red blood cell is biconcave in shape, such that a shape parameter reduced volume is closer to 0.65. We also have spherical red blood cells for which the reduced volume is closer to 1. Now, in the literature, it is not really well understood how do these different reduced volume give rise to different shape dynamics in flow. So these are the key questions I'm asking in my PhD. The first question is, how do spherical, biconcave, and tubular vesicles flow and change their shape in a steady and time-dependent flow? What is the effect of biconcave and tubular shapes on the overall mechanics of lipid vesicles in flow? How do freely suspended vesicles interact, collide, and fuse together in flow? Now, if you look at the literature of vesicle dynamics in flow, people have mainly identified three different dynamical regimes under extension. So earlier work in 2008 by the Steinberg group showed that if you have a vesicle whose reduced volume is lower than 0.6, then if you place that vesicle in a flow, it will transition into a symmetric dumbbell shape along with purling. These experiments have been complemented well by numerical simulations from AIChE group. Recently, Susan Muller group also showed that if the reduced volume is larger than 0.6, then you observe the formation of an asymmetric dumbbell shape. These experiments are also complemented well by numerical simulations from the AIChE group. Petya-Vlaska group has also shown that asymmetric dumbbell shape exists when you apply electric field on lipid vesicles. Now, most of these experiments were performed in the low flow rate regime when there is no membrane stretching. So our approach in my PhD is that I will use the Stokes trap to systematically and comprehensively study the shape dynamics of lipid vesicles in strong flows at extremely large strain rates and observe their dynamics in long time. So Stokes trap is a feedback controlled microfluidic device which allows one to control the center of mass position, orientation, and trajectories of single and multiple particles in free solution. You can see in this movie that we can control the center of mass position of a single particle. If you have a rod-like particle, then we can also control both the center of mass position and orientation of the particle. And in this movie, I'm showing that we can control the center of mass position of two particles simultaneously. This is the experimental setup of the Stokes trap device. Essentially, you have a microfluidic device which is mounted on a inverted fluorescence microscopy setup, and the device is connected with fluidic reservoirs, which in turn is again connected with a computer controlled pressure regulator. These are the pictures of the emergent flow topologies which occur in the Stokes trap device. Essentially, they are showing that we can have 0, 1, or 2 stagnation points when you try to control the position of particles using the Stokes trap device. Now, in my experiments, I will use the Stokes trap to trap a single lipid vesicle and observe its dynamics for long times. So recently, we used the Stokes trap to study the non-equilibrium flow phase diagram of lipid vesicles in flow as a function of reduced volume and the capillary number. So for these experiments, I trap a single vesicle at 0 flow using the Stokes trap. Then I slightly change the flow and observe if the vesicle is changing its shape or not. If the vesicle does not change its shape, then I increase the flow further, and I keep doing it until I reach a critical value of the flow rate at which I observe a shape change in the vesicle. By repeating these experiments over different range of reduced volumes, we saw that if your reduced volume is lower than 0.6, like in this movie, then you see that the vesicle changes its shape into a symmetric dumbbell shape. This is consistent with prior literature, and then we use that information to plot it in the form of a phase diagram. So in this phase diagram, these filled green squares represent the value of the capillary number at which the vesicle changed its shape from initial tubular shape to a symmetric dumbbell shape. And the open green squares represent the values of the flow rate at which we checked for the shape change, but the vesicle did not undergo a shape change. Then we also observe that if your initial shape of the vesicle is such that your reduced volume is between 0.6 and 0.75, then when you place the vesicle in the flow, then it will change into an asymmetric dumbbell-like shape. And these points represent in the phase diagram by means of these filled red squares. We also saw that if your reduced volume is larger than 0.75, then no matter how large flow rate you apply, the vesicle will not form a dumbbell shape. Instead, it will just transition into a stable ellipsoid-like shape. So now I will show you that how does the vesicle shape dynamics evolve in a strong flow when you apply a flow rate which is larger than the value which is required for inducing a symmetric or an asymmetric dumbbell shape change. So in this movie, you are seeing that the vesicle will change into an asymmetric dumbbell, and since the flow rate is very large, it will continue to elongate, and once you stop the flow, the vesicle will eventually recover back its initial shape. And so then you can repeat these experiments over a variety of reduced volumes, and you can plot the change in the aspect ratio of the vesicle as a function of time. Essentially, you can get the transient stretching dynamics and also the relaxation dynamics after you stop the flow. So now, if we only focus on the relaxation of these vesicles from highly deformed shapes, interestingly, we actually saw that the relaxation of the vesicle is occurring by a two-mode double exponential decay. The initial relaxation is very fast, and after that the vesicle changes into a slow relaxation stage. So if you plot the aspect ratio of the vesicle as a function of time on a semi-log scale, you get two different slopes. And we see this trend for vesicles with different reduced volumes. And we can fit a double exponential relation of this form, and you can obtain these time constants tau1 and tau2. Interestingly, we saw that the fast relaxation time scale tau1 corresponds to the membrane bending time scale, and the slow relaxation time tau2 corresponds to the membrane tension time scale. Interestingly, we also saw that if you normalize the fast time scale with the bending relaxation time, and you plot it as a function of the reduced volume, the value of the normalized fast time scale reduces with the increase in the reduced volume, essentially meaning that the vesicle will relax faster if it's less floppy. Now, using the Stokes trap, one can also expose the vesicle to a series of different levels of flow rate and obtain the steady state value, the steady state extension of the vesicles. So at higher capillary number, the steady state length of the vesicle will higher, and you can repeat these experiments at a variety of different reduced volumes, and you can obtain the steady state aspect ratio of the vesicle as a function of capillary number. So if you focus on these blue stars, up to this point, the vesicle did not form an asymmetric dumbbell, and after it formed an asymmetric dumbbell, this is the steady state shape. When you increase the capillary number, the vesicle will continue to stretch, and the steady state length will be higher. We also say that the last value of the capillary number at which we can observe the steady state, we will name it as a critical capillary number CAC. And then we do these experiments at a different value of the reduced volumes. So interestingly, if you plot out the steady state aspect ratio of the vesicles as a function of the ratio of CA over CAC, where CAC is the critical capillary number at which we observe the steady state for the last time. So interestingly, we see that there is a linear relationship if we plot these values on a log-log scale, and you can do this analysis at a viscosity ratio of 1 for different reduced volumes, and you can essentially also change the viscosity ratio by increasing the viscosity of the suspending medium. And interestingly, we see that this power law exponent alpha that roughly remains the same at a given viscosity ratio for different reduced volumes. And At a lower viscosity ratio, at lambda equal to 0.1, this power law exponent alpha, it increases, suggesting that for a given reduced volume, the tether lengths are larger for lower viscosity ratios. One can also plot the value of the critical capillary number CAC as a function of reduced volume at different viscosity ratios. And essentially we see that we can obtain this power law exponent beta. And interestingly, we see that the power law exponent beta is lower at a viscosity ratio of 1, and it's higher for a lower viscosity ratio, implying that at a given reduced volume, the aspect ratio of the vesicle will diverge at a smaller capillary number for lower viscosity ratios. Now, you can do some theoretical modeling using the Helfrich theory, and you can obtain the steady state length of the vesicles as a function of the strain rate, bending modulus, and the surface tension of the vesicle. And after building this theoretical model, we can fit this theoretical model into the experimental data of the steady state length as a function of the flow rate. So we see that when we fit this model, essentially, we get a really good fit. And after repeating this analysis over the steady state data for different reduced volumes, we can essentially find out the bending modulus and surface tension of lipid vesicles as a function of reduced volumes. Interestingly, we see that the bending modulus of the vesicles is higher when the reduced volume is higher, and the bending modulus of the lipid vesicles is lower for lower reduced volumes. And we see the same trend for vesicles with different viscosity ratios. So just to summarize my talk, in my PhD, we have developed a new technique known as the Stokes trap, which allows one to trap single and multiple particles in flow. Then we use the Stokes trap to study vesicle shape dynamics in weak and strong flows. We precisely characterize the steady state stretching dynamics of vesicles at different reduced volumes, and we gave a first view of the dependence of bending modulus on the reduced volume of the vesicles. We also observed that the steady state vesicle stretching dynamics follows a power law behavior with the dimensionless capillary number. Then at the end, I want to thank my PhD advisor, Professor Charles Schroeder, my undergrad student Channing Richter, who was instrumental in helping with some of these experiments. I also want to thank NSF and PPG Fellowship for funding my PhD research, and I'm also thankful to all the Schroeder Lab members. If you have more questions about my research, please feel free to contact me on my email, and I look forward to your questions. Thank you so much.